Hello dear students. Today we are going to discuss the first unit of plant biotechnology where we will first understand the topic or the name of the unit, that is in vitro propagation methods, where in this unit you are going to deal with the different methods that we can utilize for in vitro propagation of the plants using different explants. And within this, today we are going to focus more on one of the interesting technique, that is protoplast, its isolation, fusion, and one of the application, that is somatic hybridization. So by the end of this session, you will understand what is the meaning of protoplast, how and why we should isolate the protoplast from the plant tissue, and different methods of isolation of this protoplast. So there will be many different methods with which we can proceed for the isolation of protoplast. Now after isolation, the important task comes, that is the viability check. So we are also going to focus on what are the different methods with which we can check the viability of isolated protoplast. Lastly, we will see that what will happen with the fusion of protoplasts, like what will be the result after isolation, viability check, and hybridization of two protoplasts. So let's begin with the meaning of this protoplast. So basically, if you imagine a plant cell, it contains one important content or ingredient, which is something called as cellulose. Now this cellulose makes up the hard and rigid coating outside the cell membrane of plant cell, which is nothing but called as cell wall. This cell wall helps in the protection of inner cellular material from the outside environment. Now, this cell wall, if we simply remove it from the plant cell, what it will give you is the protoplast. As you can see in the diagram, the plant cell or any plant will be consisting of different organ system. Later on, we can see there are different tissues of the plants present inside. Now these tissues are nothing but what? The tissues are nothing but the group of cells. And here the cells are connected with another cell with one important content, that is nothing but middle lamella. Now you can see in the diagram at the top what is present is the middle lamella, after which there is a presence of primary cell wall. Within this primary cell wall we have cellulose and pectin. Both of this are nothing but the carbohydrates or the polysaccharides. So these cellulose and pectin together makes up an interconnection and forms a rigid coating, that is nothing but cell wall. Now if we remove this outer coating, what will remain is the plasma membrane and an inner cytoplasm. This is our protoplast. Now after understanding what is the meaning of protoplast, we first understand what is the usefulness or what are the applications of this protoplast, and so that we will understand why we should isolate this protoplast. So let me tell you, in a transgenic system or the branch where we are trying to manipulate the different systems for interaction or for insertion of our gene of interest into the organism for different purposes, we have several different methods. So here genetic modification, that is nothing but insertion of foreign DNA into the plant cell. Now friends, we have a cell wall around the plant cell. So it will not be very easy for us to introduce the outer gene or a foreign gene into this plant cell with the cell wall. So if we simply have protoplast with us, what will happen? The cell wall barrier is already removed. So we can easily insert the gene of interest into the protoplast if we do not have the cell wall barrier. And with this method, we can also achieve a high efficiency of gene insertion into the plant cell. Secondly, a somatic hybridization. So here we can even generate a new variety of plant if we have a plant protoplasts from the two different species. Now to understand this, we can consider one of the famous example, which is pomato. So pomato is the word, or it is the species made up from two different plants, that is potato and tomato. So you can imagine a single plant that is giving you both of these vegetables, that is potato as well as tomato. So how we have achieved this? By the protoplast fusion. So simply here we are supposed to isolate the protoplast from potato plant and likely with the tomato plant. After isolation of this protoplast from both of the plants, we are supposed to fuse them together. And during this fusion, whatever will generate will be the single cell which will have properties of both the plants, that is potato as well as tomato. So with this we can generate a new variety or a hybrid plant without using the transgenic system. Lastly, if you are interested in understanding the different growth responses of the plant cell to the different metabolites, suppose you have any new drug that you have discovered, or if you have any metabolite that you have discovered and you want to understand what is the impact of this metabolite on the growth of plant, so in that case you should have a protoplast suspension with you. And if you keep this suspension in contact with your metabolite, it will let you know that what are the different aspects that you can understand, or what is the impact of the your metabolite on this plant cells or on its growth. So apart from this, there are several other applications of the protoplasts. Hence, today we will understand how can we isolate this protoplast during different methods, using different methods, as well as how we should perform a protoplast fusion to get the new variety or a hybrid variety. Now, before we know that how to isolate the protoplast, the first task comes is to choose the source, because plant has a several different organs. It has flowers, it has stem, it has leaves, roots, etc. Now, out of all the sources or out of all these organs, which you should choose for isolation of protoplast, that depends upon your application. So here you can see few of the sources enlisted, that is leaves, then callus culture and the suspension culture. Now these are the three different sources from which I can get good yield of the protoplast. But let me tell you, leaves are the best sources chosen for the isolation of protoplast, because the method of isolation from leaves is little bit easy, as well as it provides higher yield of the protoplast. So let's begin to understand with the isolation of the protoplast methods. Now in this diagram you can see a structure of plant cell. So on the outer side what we can see is the cell wall, and earlier I have mentioned that this cell wall is made up of cellulose and the pectin. Now I have said that if I remove the cell wall, what I will get is the protoplast. But how can I remove this cell wall? So there are various methods for this removal. And one of the thing that you can see in the diagram is simply to digest the cell wall using suitable enzymes. So the enzymes, as we know, they are very specific in their action. They will cleave or they will digest a specific part of the plant cell, which is a cell wall, and later on it will give us a good yield of protoplast. So here you can see a yellow colored round shaped spherical protoplast which do not have the cell wall. Basically, it is very easy to identify the plant cell and the protoplast, because if you can see in the diagram, plant cell will have a specific morphology. Why? Because it has the cell wall, which gives it a specific shape. But if you remove this cell wall, what you will end up is getting the spherical shaped cell of the plant. So you can easily see that with cell wall how your cell looks like, and without cell wall how your protoplast looks like. Okay. Now here is a flow chart showing you the different methods of isolation of protoplast. So there are basically two important methods. One is the mechanical isolation of protoplast and second is the enzymatic isolation. Within enzymatic isolation, two more steps are given or two more types are given. One is the sequential method or single step, two step, and second is the mixed enzymatic method, which is a one step or single step method. So first we will talk about the mechanical isolation. The name itself suggests mechanical isolation is what you need to use some mechanical equipment like some scalpel or a sharp knife. You will have a plant tissue under the microscope and using a specific observation and your skills, you will use a knife and you will cleave out the plant cell wall. Now, one of the important thing we must remember here is the plant cell must be present in a hypertonic solution. Now, what is the importance of a hypertonic solution? This hypertonic solution will allow the cell to remain in a maintained form when the cell wall is removed, because the cell wall, when it is present around the cell, it will exert some pressure on the cell membrane, and later on the inner cell material also exerts some pressure on the cell wall, because of which the cells are present in a normal or a maintained condition. But we are here to remove the cell wall. So what will happen? There are some chances that cell will break up or cell will burst out. And to avoid this bursting, we must use a hypertonic solution. Now here, in a mechanical method, there are some chances that our cell will degrade or it will be injured during the process of mechanical isolation. Because after all, what you are using is nothing but a type of knife or a type of sharp instrument which will injure the cell. Now second method which comes is the enzymatic method. Now you can see one of the diagram on your screen which will explain you the entire process of enzymatic digestion. Now prior to this, let me tell you, in a enzymatic digestion we have two methods, that is to use the enzymes step by step or to use the enzymes together. Now the first question comes here, which enzymes I have to use? So here I want to degrade the cell wall, and earlier we have seen that cell wall is made up of cellulose and pectin. So simply I need to utilize the enzymes which will digest the cellulose as well as pectin. So which enzymes will come into picture here? That is pectinase and cellulase. The last suffix of this word ase indicates it is the enzyme, and cellulase means it will cleave out the cellulose material in the cell wall, and pectinase will digest the pectin material present in the cell wall. So these are the two crucial enzymes we use in the enzymatic digestion method. Now we have two choices. First, use the pectinase enzyme and separate the cells from each other which are present together in a tissue. So we will get an explant which will have a connected plant cells with each other. Simply, we have to separate each and every cell from that tissue. So who will come here for doing this task? That is the pectinase enzyme. So simply you can imagine a plant tissue which is treated with the pectinase enzyme. After the treatment of pectinase enzyme, the cells, individual cells will separate themselves from the tissue. And later on, the second enzymes that we will use here is the cellulase enzyme, which will digest the cell wall of each single cell present in the suspension. So after this method or after usage of cellulase enzyme, what we will get is the protoplast. Now same thing you can see in this diagram, but in some details with respect to practical purpose. So initially what you can see is there is a plant leaf present. Okay. Now this plant leaf will have a outer covering, that is something called as epidermis. Using the scalpel, we are supposed to scrape out some material from the epidermis. Now after collecting this epidermis, we need to place it in a solution of enzymes. Now here they have used the second method, which is a mixed enzymatic method. Now in mixed enzymatic methods, it is very simple that both the enzymes cellulase as well as pectinase, we have to keep them together. No sequential appearance will be there. So both the enzymes are present in the same solution, and within which we are adding the epidermis of the plant leaf. Now for this particular mixture of the plant leaf and the enzymatic solution, we have to keep it for performing a reaction of digestion for about 24 hours. So 24 hours, this entire mixture will be present in a dark condition. Now what will happen during this entire period? The cell wall will be completely digested by our digestive enzymes. Now later on we will see that particular mixture will have a small protoplasts which are sinking down because their density will be very low, and there will be some cell debris present. So the next task comes here is separation of your protoplast from the cell degraded material or cell debris. For this, one of the best thing that you can use is the centrifugation. So simply you can see in the diagram that they have done the centrifugation in presence of some gradient. So here we need to remove the cell's enzymatic solution after 24 hours and resuspend the protoplast into such a solution which contains some osmotic stabilizer. Sometimes we can use here a mannitol solution which has 13% mannitol along with some other micronutrients. Now after adding 13% mannitol into the solution and centrifugation, you will see that some of the protoplasts are sinking down at the bottom, and we have some cell debris present within that mixture as well. So again, second step of centrifugation is required, where the particular mixture will be added up with the 21S solution. 21S is the 21% sucrose solution. Okay, so this sucrose solution also helps us for keeping that gradient during the centrifugation. After this step, what we will get is the small little protoplast present at the top portion of the tube, and at the bottom what we will have is the presence of all the degraded material or the cell debris. So with this step we have isolated our protoplast. The next task comes here is we need to see whether or not my isolated protoplasts are viable, because if I don't check the viability, I will never know that how many protoplasts within my suspension are actually living and how many of them are dead. So this task is so much important that to check the viability of the protoplast. So there are some tests or methods that we can employ here for understanding or checking the viability of the protoplast. Basically, a viability test will monitor the formation of cell wall again, or some cellular transport, or say cellular respiration, cell division. All this important phenomenon or processes indicates that whether or not protoplast is alive. So let's see some tests for the protoplast viability. First method is fluorescein diacetate staining method. Secondly, to check the oxygen uptake by the cell. Third one is the Calcofluor White staining, and fourth is the Evans blue dye. Now students, the oxygen uptake of the cells is very easy to monitor. So I will first explain the oxygen uptake and later on we will see the other three methods. Now here, simply you should take the isolated protoplast suspension in a tube and continuously monitor the oxygen level of that suspension. Now you will wonder why to check the oxygen level. So friends, here what will happen, if a plant cell or the plant protoplast is alive, it will perform the respiration. Now during the respiration, it will take the oxygen inside, and hence the lower level of oxygen indicates that yes, there is something going on, that is called as respiration. So if protoplast is doing the respiration, that means it is alive. Okay. So you can this is the way how you can monitor the viability of protoplast. Now let's come to understand the first method, that is FDA, fluorescein diacetate staining method. Now this method relies on the usage of one dye which is called as FDA dye. Just like we used the other staining methods, here we have the stain which we will put with the protoplast suspension. Now in the diagram what you can see is the two figures. One is having the protoplast without FDA stain and other is having the protoplast which are showing some green fluorescence after staining with the FDA. So let's see what is the mode of action of this FDA. Now, all the living protoplast in your cellular suspension will have one very important enzyme called as esterase enzyme. Now, this esterase enzyme can break down FDA, that is fluorescein diacetate, into fluorescent compound. Let me repeat, the esterase enzyme present in the living protoplast will break down fluorescein diacetate into fluorescein. Now what will be the outcome? All the protoplasts who are alive will show a greenish yellow color fluorescence under the fluorescent microscope. Hence you can see in the second figure here it is showing some green fluorescence of all those protoplasts which are alive. So in a figure we cannot distinguish which one is living protoplast, which one is dead protoplast, but in the second figure definitely I can mark that all those protoplasts which are having a good green color will show me the presence of living protoplast. Okay. Now let's see the second dye method or staining method, that is Calcofluor White staining. Now friends, this particular stain relies on the usage of its particular staining principle where it will monitor the presence or formation of the cell wall. We have said that the cell protoplast will be devoid of cell wall. So protoplasts do not possess the cell wall. But that doesn't mean that they never can synthesize the cell wall. So after isolation of protoplast, after certain time, they will start producing or synthesizing the cell wall again. And we are here to monitor the same thing. So again you can see two figures on the slide. One is showing you the mixture of some protoplasts together, and the second figure is showing some colored ring around the protoplast. Now why this ring has appeared? So let me tell you, the protoplast which is synthesizing the new cell wall is alive, and within this new cell wall we have beta linked glycosides. So Calcofluor White stain will interact with the beta linked glycosides which are only present in the living protoplast or in a protoplast where it is trying to synthesize the new cell wall. So whenever the new cell wall will be synthesized, the cell wall will show you some kind of fluorescent ring. Hence you can see in the B figure, the protoplast which is spherical in nature have a fluorescent ring around itself. Why this ring has appeared? Because of the interaction between Calcofluor White stain with the beta linked glycosides around the cell, around the protoplast, because of the formation of cell wall. So what we can conclude here is, if the protoplast is alive, it will synthesize the cell wall, and if it can synthesize the cell wall, it will show you a fluorescent ring around the protoplast. Okay. So this was all about the second method, that was Calcofluor White staining. The last method here is the Evan blue staining. So again here we will use a dye called as Evan's blue dye. Let me tell you, this dye acts similarly with the trypan blue dye. Means what? In a suspension you will have the protoplasts which are dead as well as protoplasts which are alive. So when you will add some amount of Evan's blue dye into the suspension of protoplast, all those protoplasts which are alive will not take the Evan's blue dye, but all the protoplasts which are dead will accumulate the stain inside, and hence you can see in a diagram that there will be a blue colored protoplast which will be considered as dead, and you can see some green colored protoplasts which are alive. So you can only count those protoplasts which are green in color under the hemocytometer, so that you can see the amount of live protoplast within or per ml of the protoplast suspension or a protoplast mixture here. Okay? So these are all the different methods of the viability testing of the protoplasts. Now after checking the viability, I know how many protoplasts within my suspension are alive, and hence now I can go for the protoplast fusion. Now let me tell you why should I perform the protoplast fusion. Consider there are two plants. Now among these two plants, one plant is having very good characteristics of say fruit test or the height of the plant, etc. But at the same time it is very susceptible to a particular disease. Now if I have another protoplast or another plant which is of the same species but it is resistant to the disease. Now what I can do, I can simply isolate the protoplast from both of these plants, and after isolation I can fuse them together. Now what is the result after fusion? I will have a single cell or a single plant which is giving me a good height of plant, which is having a disease resistance as well, and which has a good taste of the fruit. This indicates that I can merge the characteristics of the plants which are present individually in a separate plants. After protoplast fusion, they will appear in a single plant. Okay. Now, for protoplast fusion, there are again some methods. So here you can see that these methods will involve two important types, that is spontaneous fusion and second is the induced kind of fusion. Now, spontaneous fusion, the name again itself suggests that within the spontaneous method, it will be not using any type of fusion inducing agents. Now, let me tell you, fusion inducing agents are also called as fusogens. So, the name itself indicates, as it is a spontaneous method, we just should allow two protoplasts to be pushed together so that they will fuse with each other. Now you will wonder why they will fuse with each other. So let me tell you that the cell membrane of the protoplast contains the lipid bilayer, and this lipid bilayer, if two protoplasts they are coming in contact with each other, the lipid bilayer will mix up with each other, and so that they can give you a single cell after the fusion. Now this particular method of spontaneous fusion, sometimes it may happen that it will give you a fusion of two similar protoplasts. What we want is two different plants and their protoplast only fuse with each other. But during spontaneous method, there are some chances that there is a formation of homokaryon instead of heterokaryon. So homokaryon is nothing but two similar protoplasts are fusing with each other, and heterokaryon means two different protoplasts are fused with each other. We are more interested in the heterokaryon. Hence, this method is not more likely utilized. At the same time, because of this method, the protoplast may get injured during this method or whenever you will perform this. So what you need to do for this method, simply take a depression slide, and within this depression slide put a drop of protoplast from the two different plants and gently try to shake this slide. During the shaking, protoplasts will come in contact with each other and they may fuse with each other. Now apart from this, more sophisticated methods we have with us, which is a chemically induced method. Now chemically induced method means what? We should use some chemicals which will allow the fusion of two protoplasts. So here you can see one treatment, that is something called as sodium nitrate or NaNO3 treatment. Although the role of both of these ions individually is not well understood, but they are supposed to help here for the fusion of two different protoplasts. But they will not give you a higher yield of heterokaryon. Means if you use sodium nitrate, there are chances that again the homokaryon formation would occur. Second most prominently used method is the high pH and calcium treatment. So we all know that the protoplast is a type of cell membrane, or protoplast will have a cell membrane. Now there will be a charge, specific charge present on the cell membrane. This charge will be neutralized by the calcium which we are supplying in a calcium chloride solution. So simply we will have a protoplast suspension with us, within which we will add some amount of calcium chloride with a specific concentration. And when these two things will interact with each other, we will also put the second solution of the protoplast, means protoplast from the two different plants. Now, with the help of high pH as well as calcium treatment, the charge on the protoplast membrane will be neutralized, and so that the two protoplasts will easily fuse with each other. Now, the third method here is your PEG method, polyethylene glycan method. Also, this particular PEG chemical is very helpful for fusion. Why? Because if you add polyethylene glycol in a suspension of protoplast, one reaction would occur, or one phenomenon you can see, that is something called as agglutination. Agglutination here is nothing but gathering of the protoplast with each other. Means the two different protoplasts will try to come in contact with each other more and more because of this PEG, and also it acts as a fusogen where it will allow the fusion of two protoplasts with each other. Lastly, we have a electrofusion method which is highly sophisticated, and it comprises with certain events which we can see in an upcoming slide. So in case of electrofusion, basically what will happen, we are using a specific electric current in a protoplast suspension. So you will have a protoplast suspension in which you will put some amount of current, and under the influence of this current, the cell will try to make a chain. The protoplast will form a particular chain, and then as they are aligned with each other, they will later on fused by change in the brief spell of the current. So yes, in this diagram we can see that if we put two charged electrodes in a protoplast suspension, what will happen, that the protoplasts present inside the suspension will align themselves in a chain form. And why this will happen? Because we have provided a low voltage electric current. There is a phenomenon called as dielectrophoretic collection of protoplast. Dielectrophoretic here means both of the charges are present in a same solution, and electrophoresis means under the influence of electric current, they are trying to move from one place to the other place. So similarly here what we can see is there is a formation of chain of the protoplast in a suspension. Now you can see small black circles in a D diagram with some charge, and they have aligned themselves in a chain form. Now let's see what are the different molecular events that will occur during this electrofusion. So we have seen that protoplasts have formed a chain during the electrofusion. Now in this A diagram you can consider two protoplasts are very close to each other, okay, and there will be having some lipid bilayer of both of this protoplast. Now this is the result of electrofusion, where we have initially provided a low voltage AC current. Now you can see some change in the B diagram. In B diagram we can see that the lipid bilayer from both the protoplast or the cell membrane are intermingled with each other, or they have intermixed with each other. This will be the result of change in the AC current, change from AC current into the DC current. So suddenly we are going to change AC current into the DC current, as well as we will increase the voltage slightly. What will happen as a result of this change is lipids from both of the protoplasts will mix with each other, and they will appear like in this blue figure. Lastly, after again changing in the current from DC to AC, you can see in the last figure that the protoplasts have realigned themselves. but this alignment is different from the A. In A figure, these are vertically aligned, that two protoplasts are different from each other. But you can see in the last figure, the protoplasts or those lipid bilayers are vertically aligned, which indicates that there is a complete equal distribution of the two protoplast content which were separate initially, but after electrofusion, both of them or both of the protoplasts are now mixed with each other equally, giving you a hybrid cell. So students, this was all about what we have studied in this entire lecture, that what is the meaning of protoplast and how we can isolate. So what you people are going to do as an assignment, you have to check all the methods of protoplast viability, including those that I have taught you in the lecture, and apart from this, if there are any other methods of checking the viability of protoplast, you will find them out and you have to elaborate each of the method in detail along with some diagrams. So here what is expected? First, enlist all the methods of viability check. Later on, involve the description of every method in detail in the assignment. Secondly, you will also write a comparative analysis of different methods of protoplast isolation, which was the mechanical method of isolation and second was the enzymatic method of isolation. Along with there some advantages and disadvantages. So with this part, you will be finishing up with your assignment. Let's summarize what we have seen in today's lecture. First of all, we understood the meaning of protoplast and why there is a need to isolate this protoplast. Like there were the applications such as in the genetic modification, then in the somatic hybridization and in the understanding of growth parameters. So with this all, we have understood the meaning and need of protoplast isolation. Later on, we have seen what are the different methods by which I can isolate my protoplast from the given tissue. So there were different sources, leaves were there, callus culture was there and suspension culture, out of which I have explained in detail you, that is from leaf how we can isolate the protoplast. And lastly, we have also seen what are the different tests for checking the viability of protoplast, and lastly how we can fuse the two protoplasts for a purpose of somatic hybridization. So we have seen both the meaning of protoplast fusion and what will be the result after fusion, which is nothing but formation of hybrid plant, which is a result of fusion of two protoplasts. Now, in the upcoming lecture, you will be understanding after fusion of protoplast, how you will identify which one is your heterokaryon. So during today's lecture, we have discussed that there are possibilities of the formation of homokaryon as well as heterokaryon during the protoplast fusion. So here, what we are more interested in is in the heterokaryon. So how you will come to know that whether or not your suspension contains homokaryon or heterokaryon. For that, we need some selection methods. So this selection of fusion products we will understand in the upcoming lecture. Secondly, we will also see that after selection, how can I separate homokaryon from the heterokaryon material. Then we will verify that whatever we have isolated is really a hybrid or is it really a heterokaryon or not. So that will be included in the verification and characterization of somatic hybrids. Lastly, we will see one of the related concept, that is something called as hybridization or formation of two cytoplasmic hybrids. And we will also come to know about the application and limitation of this technique called as somatic hybridization. So all these contents will be included in your upcoming lecture. You can see some of the references I have put down here, which you can use for the further reading of all these methods and contents that we have completed today. Thank you.